Today we'll talk about the blood group in humans. In humans, there are four blood groups: A, B, AB, and O. Humans have four blood groups: A, B, AB, and O. This blood group A, B, and O, it was first discovered by Landsteiner. And blood group AB was discovered by De Castillo. These four blood groups A, B, AB, and O, they are classified on the basis of antigens or glycoproteins present on the surface of RBCs. So we have these four blood groups. Blood group in humans are controlled by three alleles. They are controlled by three alleles: IA, IB, and small I. These IA and IB, they are codominant alleles, whereas this small I, it is recessive to both IA as well as IB. So four blood groups AB, and O in humans are controlled by three alleles, and they will form how many phenotypes? A, B, AB, and O. These are phenotypes. Four phenotypes we can say. And how many genotypes should be formed? 6 genotypes should be formed: IA, small I, IB, and IB small I, IB, and small I. So we have 1, 2, 3, 4, 5, 6 genotypes. Regarding the blood group A, if we talk about the blood group A, if we'll talk about the blood group A, we'll find that this blood group, it is controlled by this blood group. It is a phenotype. It is controlled by genotype IA or IA small I, because small I, it is recessive to both capital IA as well as capital IB. And the antigen which is present on the surface of A blood group individuals, that is, they have A antigen present on the surface of, this is RBC, this is antigen, A antigen on the surface of RBCs, and they have B antibodies. They have B antibodies present in their blood plasma. Then we have, this is blood group 1, then blood group 2, blood group B. Phenotype, their genotype would be IB, and IB small I. And B antigen is present on the surface of their RBCs. This is B antigen on the surface of RBCs. They have got B antigen on the surface of RBCs, and they have anti-A antibodies present in their blood plasma. Anti-A antibodies. They have anti-A antibodies present in blood plasma. Same is the case, we have third one, blood group AB. AB, it is having genotype IA, IB. It is having genotype IA, IB, IA and IB, and they have A, B antigens on the surface of RBCs. That is allele IA and allele IB, they are codominant. That is why they have both antigens on the surface of RBCs, A antigen as well as B antigen on the surface of RBCs. And this blood group AB, they have neither A antibodies nor B antibodies in the blood plasma. So they do not have antibodies, neither A antibodies nor B antibodies in their blood plasma. So they are universal recipients. Then we have fourth blood group, that is blood group O. The genotype is small I, small I. That means small I means no antigen on the surface of RBCs. Antigen absent on the surface of RBCs. So they have anti-A and anti-B antibodies in their blood plasma, present in their blood plasma. As we have seen these blood groups, blood group A, blood group B, blood group A, B and O, we have also seen there what are the genotypes and antigens. Among these, this blood group AB, it is called as universal recipient. AB blood group individuals, they are universal recipients. Why they are universal recipients? They can receive blood from A, B and O because they have no antibodies present in their blood plasma. Whereas blood group O individuals, O blood group individuals, they are universal donors. Why they are universal donors? Because they do not have antigens, no antigen on RBCs, because they do not have. So AB blood group, these are AB blood group individuals are universal recipients. They are universal recipients. You can receive blood from all the four blood groups, and they are universal donors. O blood group individuals are universal donors. And we say O negative. O negative. O negative means that this is the best recipient. O negative, this is the best donor. O negative is best donor because the antigens are absent. O negative, that is donor, best donor. O negative means antigen is absent. And sometimes we write AB positive. Positive means antigen present. So we can say that this O negative means antigen is absent, and this blood group O individuals, they are best donors. So blood group O individuals are universal donors, and blood group AB individuals are universal recipients. Then we can show it like this. It means that we have A blood group. This A blood group, it can receive blood from A as well as O, because O is universal donor. It can give blood to A. It can give blood to A as well as to AB, because AB is universal recipient. Same is the case. This O is universal donor. O blood group is universal donor and AB is universal recipient. Same is the case, we have B blood group. B blood group, it can get blood from B individuals as well as O individuals, because O individuals are universal donors. It can give blood to B individuals as well as to AB, because AB is universal recipient. Same is the case, we have AB. This AB blood group, it is universal recipient. It can receive blood from A, it can receive blood from B, it can receive blood from AB, and it can receive blood from O, because it is universal recipient. And it can give blood to AB only because it has having anti-A, because it is having antigen A and B on the surface of RBCs. Now we have O blood group. O blood group. This O blood group, we know that it is universal donor. It can get blood from O individuals. It can give blood to all the 4 blood groups, because it is blood group O is universal donor. It can give blood to O. It can give blood to O, A, B and AB individuals. Now we have shown that different types of blood groups. There is a term, so we can say that this is blood group AB, it is universal recipient, and blood group O, it is the universal donor. There is a term, that is RH factor. What is RH factor? What is RH factor? This RH factor, it is simply an antigen. Yeah, we can say that glycoprotein, which is present on the surface of RBCs. This RH factor, it is an antigen or glycoprotein present on the surface of RBCs, and it was first discovered by Landsteiner in the Indian rhesus monkey. And about 93% of Indians have RH factor present on the surface of their RBCs. So this was regarding about their blood groups. Then we have another important thing, that is what happens, why it is necessary during the blood transfusion to determine the compatibility of the blood. So it is necessary to determine the compatibility during blood transfusion. during blood transfusion, it is necessary to determine the compatibility of blood groups. For example, if we have A blood group individual, it should receive blood from A or O blood group individuals. Now, if in the A blood group individual, suppose B blood group has been injected, then what will happen? There would occur an antigen-antibody reaction, and there would be agglutination or clumping of RBCs. So in order to prevent this clumping of RBCs or agglutination, it is necessary to determine the compatibility of blood groups during blood transfusion. For example, we have suppose we have A blood group individual. This is A blood group individual. This is A group individual. This is A blood group individual, and obviously it has anti-B antibodies, and it has A antigen on the surface of RBCs. Now, if in this A blood group individual, we will introduce what? B blood group injected. What will happen? This B blood group individuals, obviously it has what antigen? It has B antigen on RBCs. So these anti-B antibodies and antigen, these antibodies of A blood group, they will interact, they will react with these B antigens, and it will cause agglutination of the RBCs or clumping of RBCs. There would be clumping of RBCs, clumping or agglutination of RBCs. That means RBCs will get collapsed. It will be like this. This is the mismatching of the blood groups. So this occurs during, this clumping occurs during mismatching of blood groups during transfusion. Here we see this is no mismatching, no clumping, no agglutination. So it is necessary to determine the compatibility of blood groups during transfusion. So this was regarding the blood group in humans, which is universal donor, which is universal recipient. AB blood groups are universal recipients, and O blood group are universal donors. O negative means antigen is absent on the surface of RBCs in O blood group individuals. AB positive means antigen present on the surface of individuals with AB blood groups. Then if there is, in the blood group B individual, A blood group is introduced, there is agglutination or collapsing of the RBCs. So in order to prevent this, it is necessary to determine the compatibility of blood groups during blood transfusion. Then we are discussing about RH factor glycoprotein, which is present on the surface of RBCs, and about 93% of the Indians have this RH factor in their RBCs. Thanks.